All right, welcome back today, and today we're going to talk about the plasma membrane and its structure, so the parts that make it up, and its function, so its role as being a semi-permeable barrier and how that semi-permeability arises. So we'll get started here, and first you can see a little artistic representation of the plasma membrane right there. So the plasma membrane is what we call a fluid mosaic. I'm gonna break that down. It's fluid because it's pliable. We can move it around easily because it has a lipid component. The phospholipids in the membrane can move laterally. But it's also a mosaic, which means it's made up of a combination of different molecules like lipids and proteins and carbohydrates. So it's a fluid mosaic model for that reason. Now, the primary part that makes up a plasma membrane is the phospholipid, like we talked about last unit. And here is just a representation of a phospholipid kind of in a artistic way. And this is more like a ball and stick space filling model. All right. So here in both is the phosphate groups and then the fatty acid tails in both, just so we can see which is which. So there's what we call a phosphate quote unquote head, and we pointed that out. And it's hydrophilic because it is polar, so it mixes in water, it likes to be around water, it dissolves in water, attracted to water. And the fatty acid tails are hydrophobic because they're nonpolar, they don't have any of those electronegative elements. They're repelled by, they don't like water, they want to be as far away from water as possible. So when we um, assemble them into a cell plasma membrane, they arrange naturally into a bilayer. So bi means two, a layer composed of two phospholipids across, like this. So you can see right here, this is the bilayer. All right, because the phosphate heads are hydrophilic, they are facing the extracellular fluid and they are facing the inner cellular fluid. So this is the water outside of the cell and the water inside of the cell. And so the hydrophilic parts, the head parts, will be closer to that. And the hydrophobic tails will be all scrunched up in the middle, as far away from the water as possible. And that's why they kind of spontaneously form this membrane or this bilayer, just like that. So they orient themselves like this. So here is another bigger picture of the phospholipid bilayer with the heads here and here, and the tails in between. Okay. And then there's water on the outside and on the inside. So the polar hydrophilic heads on both sides and the non-polar hydrophilic tails in the middle. All right. Because of this arrangement, it serves as a barrier or a border separating the outside and the inside of the cell. And because they're composed of this big area in the middle filled with the non-polar hydrophobic tails, things that are non-polar can easily pass through the membrane, like lipids, but things that are polar can't, like sugar and water. and salts are other ions. So, the phospholipid bilayer is impermeable, meaning that it can't be crossed by polar molecules. I think what should have been here is the word large. Large and polar molecules can't fit through here. So sugar is polar, so it can't cross. Water is polar, so it can't cross all by its own. Salt is polar, so it can't cross all on its own. But it is permeable to small and nonpolar molecules, like lipids, or like oxygen, or nitrogen, or carbon dioxide. These things are small, nonpolar, and so they can easily cross the plasma membrane in the case of oxygen, or out, like carbon dioxide and nitrogen would want to get out of the cell. So that's going to be important to remember, that it can't be permeated, it can't be crossed by large or polar molecules, but it can let in small and nonpolar molecules. So the cell membrane, like we said, separates living cells from their watery environments outside and inside. They're about 8 nanometers thick, and then they are really responsible for controlling the traffic in and out of a cell. So they allow some substances to cross more easily than others. Small, nonpolar molecules cross easily. Large or polar molecules cannot cross. But how do we get some of the polar molecules in and out of the cell? Like sugar, we need to get into the cell to be used for energy. The membrane becomes semi-permeable through protein channels. So here are protein channels that are embedded in the plasma membrane. And here's kind of like a more accurate drawing of it. And they allow specific material to cross the cell membrane under specific circumstances. So they're kind of like a doorway or a portal for those things to cross when they're able to. So like sugars or some amino acids or water, salts, other waste products can go in or out of the cell when they couldn't naturally get through the plasma membrane. Membrane proteins, like we talked about, determine the membrane's special function. So cell membranes and organelles have unique collections of proteins, and some of them in the cell membrane are called peripheral proteins, like you see over here, that are loosely bound just to the surface of the membrane. Kind of like if you think about like the antigens in a red blood cell, they identify or are identity markers to say, hey, this is this type of cell. Hey, body, this is me. I'm not a foreign invader or a pathogen, like those. There are integral proteins that penetrate the bilayer that go to both sides. And these are like the transmembrane proteins that we talked about that allow polar substances to pass through the membrane. Um, and they are really responsible for a lot of the homeostatic functions of the cell. So like those right there. All right. I am just going to skip ahead here to the next thing I want to talk about. On some of the proteins that you will find in the cell membrane, you'll see that they have carbohydrates attached to them. And these carbohydrates play a key role in two cells recognizing each other. Whether it's two blood cells or a blood cell and a liver cell, they play a key role in recognition. And it gives the ability to cells to distinguish one cell from another and allows cells to stick to each other. So, for example, if you get a cut and cells need to replicate to repair so that they can stick to one another So you don't just have like this hole right where your cut was and it never gets better. Um, and another important thing for cell-to-cell recognition is that it plays a key function in the immune system. This is how, for example, white blood cells recognize foreign invaders, bacteria, pathogens, and are able to attack them specifically, and not your own cells. Okay, so how your body can tell that, hey, cell is part of me, it's good, versus that cell is not part of me, it's bad, we need to attack it and kill it, it could cause us harm. One of the other things that's important in the cell membrane is the cholesterol molecule. Um, and it helps the plasma membrane maintain its fluidity. So, for example, at high temperatures, cholesterol stiffens the plasma membrane so that it doesn't become all jelly-like in that high temperature and completely lose its stability. But at low temperatures, it prevents the membrane from freezing. So it helps to regulate the plasma membrane's fluidity, not to become too soupy and not to become too rigid. And so cholesterol is found kind of embedded, spaced out between the phospholipids to help maintain that fluidity. In animal cells, the area in between cells is called the extracellular matrix, and it contains various amounts of different protein fibers and complex carbohydrates right here that help to assist in the communication between cells. So in this picture, if you look at all this stuff right here, that's the extracellular matrix, and so it has generally that function. Um, so here is a picture of kind of what we've been talking about. So we have the channel proteins, those integral proteins, some peripheral proteins on the outside. Here's the cholesterol maintaining the fluidity of the plasma membrane, and those carbohydrates that are sticking out to help in cell-to-cell communication and recognition. And then out here, the extracellular matrix. And there's the cytoplasm inside the cell. So you can see with all the labels the different things that we've kind of talked about. So just to review, the plasma membrane is made up of the phospholipids with their polar hydrophilic heads and their nonpolar hydrophobic tails. On the outside, the extracellular matrix resides in animal cells that contain various protein, fibers, and complex carbohydrates to assist in cellular communication. We have the carbohydrate chains that only occur outside the surface of the cell membrane to aid in adhesion between different cells that act as receptors for cell signaling and also with cell-to-cell recognition as well. And then we have cholesterol, which is a lipid that's inside the plasma membrane that helps to maintain its fluidity in different temperatures. And then different proteins, integral and peripheral proteins that serve many different functions, but one of the important ones is as channels to allow polar molecules in and out of the cell that are necessary to maintain homeostasis. So that's going to be a bit about it for this video. Feel free to go back and watch it as many times as you need or ask me some questions.